Okay, ladies and gentlemen, hope you're all good. So, I am going to very briefly give you guys a summary of what was discussed in class. I know that some of you had some issues logging into the platform to take the class today. So, what I'm going to do is I'm going to take a couple of minutes and just kind of go over the most important things that were discussed. So, I'm actually sharing my whiteboard so that way if there's anything that is relevant that I think you guys should take a look at, I'll just open up a text box and we can start. Okay. So today's topic was on section 1.4 of your textbooks. And the topic is biologists, tools and technology. So, in this section, we're going to talk a little bit about the tools that biologists use to recollect data, some of the more basic instruments. And I'm actually going to go ahead and go directly from your textbooks. And I'm on page, I believe it's page 16 or 17. Um, and I'm going to read a little bit or give an introduction. It says, until the late 1600s, no one knew about cells or single-celled organisms. The microscope before the microscope was invented. Scientists suddenly had the ability to bring living things into a different perspective. So, up until maybe the 1600s, there was not a lot of tools at the disposal of scientists to study more of the microscopic universe, right? And I mentioned in class this morning that there were the theories of humors by Hippocrates, and he was is called the father of medicine, and he was a doctor, and what he believed was that people's moods and their health depending upon their bodily fluids. He would dissect the corpses, and he came up with a theory of humors based on this and other things, and that theory of humors lasted for centuries. And it wasn't until in that era, the 1600s, when people started to make more scientific discoveries that the microscope was invented. Now, the microscope is not the only tool, but of course, when we talk about studying single-celled organisms or bacteria, it is the tool that comes to mind. So, if we go to your book, it says that microscope basically provides an enlarged image of an object. So, basically, a microscope provides an enlarged image of an object. Okay. So essentially, a microscope, and in fact, when the microscope was invented, it was kind of like putting lenses on top of lenses on top of lenses. A little bit later on, when we start talking about the cell theory, or when we start talking about microscopy, and we'll go into the story, more specifics of how it was invented. But pretty much, it was almost an accidental discovery. Okay. It says that the first microscope magnified object, but did not produce clear images. Okay, so that's very important to understand. When the microscope developed, they didn't really produce a lot of clear images. And it says that by the 1800s, most microscope had combinations of lenses that provided a clear image. So it wasn't until the 1800s um, that microscope actually produced more clearer image and clearer, excuse me, images. Okay, so it wasn't until about the 1800s that microscope had a better resolution, because the first microscopes were pretty much just kind of like lenses grouped on top of each other. And although they helped, they didn't really become more clear until the 1800s. So even though the microscope was invented in the 1600s, people still used the theory of the humors for a couple of more years. And even there was the belief, and I mentioned this in class, that living things came from non-living things. Like they would leave a piece of, like I mentioned in class, they would leave a piece of meat on a counter space, and all of a sudden it would be full of maggots and flies. And some the scientists at the time just believed that all that was just the dead or rotting, or the dead corpse producing life. So it says that the first microscopes, or around that time, the light microscopes, um, magnified about 1,500 times their actual size, and samples are often stained with chemicals to make details. So the light microscope, and this is the microscope that you'll find generally in the classroom. It says that a light microscope, and that doesn't use any electricity or electrons or anything like that. A light microscope can magnify an object about 1,500 times their actual size. And of course, these are not the only types of microscope that we have, and we're going to talk a little bit about the electron microscope. Okay, so the electron microscope is much more modern than the light microscope, and the electron microscope wasn't developed. The electron microscope wasn't developed about until about the 1950s. And interestingly enough, and I mentioned this in class as well, and when we start talking about the genetics portion of the section, we'll probably mention it again. But the double helix, or the actual structure of the DNA, which I mentioned in class was kind of like a ladder, wasn't discovered until the 1950s. And that was about the same time that the electron microscope was developed. Now, I'll go a little bit more in detail in that a little bit later on. Now, continuing to talk about the electron microscope, it's called the electron microscope, and the reason it's given that name, as you may suggest, is because it uses beams of electrons instead of light to magnify an object. Okay, so electron microscope use beams of electrons instead of beams of light, which is what the light microscope uses. That's why it has that name. Okay, so there are two types, and I'm actually going to go ahead and clear the images. Okay, so just kind of going back real quick to what we started. We're talking about the tools that help biologists, and we talked about the microscope. And the microscopes were invented around the 1600s, but it wasn't until the 1800s that more clear images were produced, and that a light microscope can about magnify about 1,500 times the actual size of an object. And we're talking about the electron microscope, which wasn't developed until about the 1950s. And it's called an electron microscope because instead of using beams of light, it actually uses beams of electrons. So I'm going to clear my drawings here, and then we're going to continue uh, to talk a little bit about the microscopes. And another important fact about the electron microscopes is that they can actually magnify an object to about 100,000 times their actual size. So electron microscopes produce clearer images up to 100,000. I'm sorry, guys. 100,000 times their actual size. It's about 100,000 times their actual size. All right. So now I mentioned this in class, but electron microscopes, they aren't necessarily used to study live specimens, although you could, if you create some sort of a vacuum. And in case of a vacuums, you could be coating the specimen with kind of like a metallic thinner. And what that does is that it'll be have the electron have the electrons kind of bounce off of it and produce an image. Um, they can be used to directly study uh, proteins or molecules. Okay, so let me type that down. Sorry, my keyboard froze a minute. As I was saying, um, electron microscopes can be used to study a specific um, protein or an individual. I'm sorry, that's the word I'm looking for. An individual protein or molecule. So I can't necessarily study a living specimen, but I can study a specific protein if I'm studying cell growth or anything like that, or some researchers may even use electron microscopes in terms of like studying cancer cells or cells that undergo a specific mutation in a specific protein. So can be used to study an individual molecule or protein. Okay, can be used to study an individual molecule or protein. And there are two types of microscope. We have the SEM, or the scanning electron microscope, and we have a TEM, which is a transmission electron microscope. Okay, and it's actually the the TEM, or the transmission electron microscope, that makes a two-dimensional imaging. Okay, so types of microscope. Okay, types of electron microscope are the SEM, or the scanning electron microscope. And the SEM actually forms uh, a three-dimensional image. Okay, and it says that it scans the surface of a specimen with a beam of electron. Usually the specimen's surface is coated with a very thin layer of a metal. So it's actually in the SEM microscopes that you kind of coat the specimen with a type of thin layer of metal that helps the electrons bounce off the beam. Okay, um, and forms a three-dimensional image. Forms a 3D, or three-dimensional image, and specimen is coated with a metal, or a thin metal layer. Okay, is coated is coated with a thin layer of metal. Thin layer of metal. And again, what that does is it helps the electrons bounce off of it, and it forms a 3D image. Now, the other that we have is the TEM, or the it's called, or the transmission electron microscope, and this one makes a two-dimensional image. Okay, and it's kind of like a light microscope, I guess, because it's smaller, or not small. I'm sorry. It only makes a two-dimensional image, but of course, a TEM or a transmission electron microscope produces a much larger image. Okay, so TEM produces a two-dimensional image, or 2D image. All right. So those are the electron microscopes. Now we're going to talk a little bit about medical imaging. Okay, so I'm going to go ahead and clear this. Now, medical imaging, ladies and gentlemen, are basically X-rays and MRI machines and even sonograms in the case of women that are pregnant. Um, but here is more or less a definition of at least of one. Okay, so medical imaging. Let me write that. Medical imagery. I'm sorry, or imaging. And these are X-rays, MRI machines, and although this one is not in your book, we have ultrasound, which is also used for medical imaging. And ultrasound just basically turns sound energy into imagery or into an image. So it's called a sonogram because, for example, in the case of women who are pregnant, it actually, or for whatever other reasons people have to undergo ultrasound, and it kind of, and this is me being very simplistic, it kind of turns sound energy into an imagery. Okay, so those are examples of medical imaging. We're going to talk a little bit about the X-ray real quick. The X-ray is, I want to say, the oldest, but I would say it was probably one of the first medical imaging tools that were used. I mentioned in class this morning that way back when X-rays started to first be used, they would even use X-ray or take X-rays of women who were pregnant, and you can even go and look at some old rare pictures of an X-ray of a pregnant woman. So X-rays um, is again formed by X-rays, an image formed by X-rays, because I was explaining in class that X-rays is found in nature. It's not something that was invented, and I talked a little bit about the spectrum, but it's an X-ray image is basically formed when an X-ray passes through soft tissues. Okay, so an X-ray image is formed when an X-ray passes through um, soft tissues and the skin, and is absorbed by the bones. So that's why X-rays are used when someone, like, mainly, not the only reason, not the only time, but mainly X-rays are more commonly seen being used when there's a suspicion of some sort of fracture, because the bones are thick enough to actually absorb the X-ray, and that helps the machine create the image. So the other that we have is MRI, or magnetic resonance. But let me see if there's anything else important about the X-ray. Um, and of course, the bones and the teeth, because they are hard structures. And again, it's useful for studying bones or looking at the skeleton to see if there's some sort of fracture somewhere, or in terms of the teeth, if you're, for whatever. dentists need to get the x-rays of, sometimes to even see cavities or something like that. And it's because the teeth and the bones are very hard structures, and they can absorb the x-ray and help produce an image. The other that we have is MRI. Okay, and MRI imaging is called magnetic resonance imaging, and MRI. And an MRI, it says that uses a strong magnetic field to produce a cross section image of a part of a body. So uses magnetic fields. Okay? MRI uses a strong magnetic field. And what the strong magnetic field, or MRI, does is it produces a cross section image. So it's kind of like if they were taking the images and cutting it in half. So it produces a, to produce, I'm sorry, to produce a cross or a cross section image. I'm sorry, a cross section image of a body part. Cross section image of a body part. Okay, so it just kind of takes an image and crosses it in half, and they're used to study ligaments, and there's even an fMRI. Okay, and an fMRI or brain scan. This is another one. It's commonly called, or it's, some people may refer to it as a brain scan, or fMRI is used. Okay, and fMRI is is used. It's also called functional MRI. Uh, it's used to show which areas of the brain are active. It's used to show which areas of the brain are active while a person is doing a particular task. Doing a particular task. Okay. So you have two types of MRIs, or not two types, because it's not a different type of MRI. It's not a different type of MRI. It's actually an MRI technique. We have the fMRI, and both use the magnetic fields or strong magnetic fields to create an image. And actually, MRIs and fMRIs are much safer than using X-rays, because an overexposure to X-rays can actually cause mutations in the cells. And in the later years, it was discovered that that X-rays could actually cause mutations and lead to cancers. So that's why they don't use X-rays to study, or to obtain an image of a pregnant woman. Um, the last thing we're going to talk about is molecular biology. Okay, now to talk about molecular biology, like I mentioned in class, and over the last since the beginning of the 21st century, more or less, and in the last, I would say, 20 to 30 years, medicine has taken a more molecular role. And not just medicine, but we have a study in the terms of in the criminal justice field. We use molecular biology and gene technique to study the scene of a crime, or all these other things that have the scene of a crime, to see if in fact there was a someone present that could have contributed to it. They're used in the preparation of transgenic foods. So in the last 20 or 30 years, molecular biology has really taken a very different, or a very active role in shaping our reality. And we're going to define what a gene is. And a gene is basically a segment of DNA. Okay, so a gene is a segment of DNA that stores information. So all of the information is in our genes, and our genes are just a segment of DNA. And I think I mentioned that it's thought that DNA can actually, or an equivalent of a strand of DNA, can have one strand of DNA can have or a gene can have the equivalent to about 1,000 pages worth of information. So um, it's a very complex molecule to study. Um, and again, these have led to a lot of developmental technology. Understanding genes has led to a lot of technological advances, like, for example, genetically modified food. So, um, GMOs, or genetically modified food, are a result of understanding genes. We have transgenic plants and animals, and the replacement of faulty genes. So they can also be used in the replacement, or understanding molecular biology can be used in the replacement of faulty genes. Okay, so in the replacement of faulty genes, the replacement of faulty genes. Um, and that has actually led to an entirely new brand, or a branch, excuse me, of biology called genomics. And genomics, which I'm going to define right now, genomics is the study of genomes, both within and across species. So genomics the study is basically the study of genomes, the study and comparison of genomes. Study is a study and comparison of genomes. And again, genomes is the study of genes, and a gene is a segment of DNA. And I mentioned in class that the Human Genome Project was a project that set out to study the entire human genome to understand how certain conditions are acquired, or to understand what codification the DNA has. It's kind of like breaking the code of DNA. So DNA is arranged in sequences, and let's see if I can illustrate that real quick. So basically, the Human Genome Project wanted to study DNA. So DNA is a double helix. It's referred to as a double helix structure, and it's usually, at least in our cells, in the spiral form. But you can see it mainly projected as kind of like a ladder. And the Human Genome Project set out to study every single gene in humans, because these little bars that you see here are actually nitrogen bases. And these nitrogen bases contain an arrangement, and that arrangement codifies for different things. Hair color, eye color, skin color, etc., etc. And certain diseases or malformations are caused by problems here, and the Human Genome Project set to study this out, because they believe that cracking this code not only would help us better the quality of human beings by maybe having better medicine, but also help crack the code of evolution. So that's how that gave way to the development of genomics. Now, one very important thing, and I'm almost done with this, is that all of the information from genomics is managed by computer databases. So when the Human Genome Project was underway, or the studying of DNA, basically they report all these findings or all these sequences into databases, and these databases are used by scientists all over the world, because we know, as we studied in the previous section, we have to, or scientists share their information worldwide. Now, all the information in from genomics is or genomics, excuse me. All the information from genomics is managed by computer databases. Okay, so all the information of genomics, all the information in genomics, genomics is stored in computer databases. Okay, and actually accessing this information is called data mining. Okay, so accessing this information through a process, we can say through a process called data mining. A process called data mining. Okay, so basically, genomics is a study of genomes, not just human genomes, but you can also study genomes of other species, and animals and plants and so on and so forth, and even bacteria. And the information that has been obtained, in the study of the genomes is in a database, and scrolling through the databases to find the different combinations is a process called data mining. So this last topic of the section basically tells us that biology, or the study of molecular biology, has in fact, given biologists more tools other than just microscopes, or other or electron microscopes, but that in this new 21st century, and especially in this era that we're living in, it has provided with a plethora of tools that help biologists better understand not just us as humans, but in general the environment, or the biosphere that we live in. And for example, and I'll give you another one that's not in your book, but there were even long ago, there was talks of creating bacteria, for example, that could actually help with oil spills, because they thought they could genetically modify the bacteria, and it would have an enzyme, and we'll talk about that later on, that could help actually break down the oil components, and that's one way that molecular biology, or can help us protect our environment as well. So, ladies and gentlemen, this is the summary of section 1.4. Hopefully this will help, and I will see you guys in the next class. Bye.